ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Hello students, let us see that last part of transport in plants. So far we have discussed major concepts and most important concepts of transport in plants. Now here and there if you look at, we have some more extra points are needed which are very much essential for you all to remember. Now let us concentrate on the last class we studied about the numericals. In the final unit paper we have chance of getting the numericals, so try to practice all that different pattern of numericals. Right. So in transport in plants, let us look for very important extra points. So when you talk about starting from the part like guttation, or you take osmotic pressure, when you take into consideration, where you find high osmotic pressure, that is we find only in case of halophytes. So we find high osmotic pressure we find in case of halophytes. Halophytes are nothing but the plants which grow in highly saline soils. Where do they find? These are the plants which find growing in high saline condition, meaning high salt concentration. So these are the ones which contain high osmotic pressure. Right. Now apart from this, when you talk about high osmotic pressure, then even you can take into consideration of, when you talk in terms of osmotic pressure, water potential also can be calculated as osmotic potential plus pressure potential So for the last class we have done the numericals. And sometimes in the numericals they may not give solute potential values, sometimes they will give osmotic potential values. That means osmotic potential itself can be referred as psi phi plus pressure potential value itself is called as psi p. Okay. Osmotic potential is nothing but psi pi, itself is also called solute potential. Sometimes solute potential values may not be given, solute potential terminology may not be used in the question. At that time they make use of the concept called osmotic potential. Osmotic potential plus pressure potential, that constitutes what is called as water potential. Right. So whenever you take into consideration of, let us talk about high rate of loss of water by guttation. You take high rate of loss of water if it is by guttation. High rate means you can take the best one, that happens in case of colocasia. Why? Because colocasia is nothing but what? A herbaceous plant, right? Similarly, we have some instruments like if you take photometer. Photometer is used to measure transpiration. Photometer is an instrument, generally it is used to measure transpiration. Similarly, we can also take manometer. It's an instrument is being designed and this manometer is used to measure root pressure. Okay, so we have to remember. Similarly, if you look at other concepts, we have discussed even guttation, right? So even we studied that guttation happens to minute pores which are present on the veins, which we call them as what? Hydatodes. These are present where? Minute openings present on veins, and these veins are always open. They always open, but loss of water happens either in the early morning or late in the night by the process called guttation. Generally they always open. When atmospheric humidity is more, atmosphere is not taking water, then water comes out even from veins, because so much of accumulation of water, because early in the morning, late in the night, atmospheric humidity is more. Atmosphere not tend to take the water, all mesophyll cells are fully filled with water, then water tend to come out from the veins. Generally hydrothodes are always open. That is what we studied in case of guttation, right. Let us see some more points. Now if you take plasmolysis, plasmolysis experiment was demonstrated, plasmolysis experiment demonstrated in the plant name called Tradescantia. In cytology first year also, in cytology we studied about a few scientists and what they have discussed, what they were discovered. At the time also we discussed about what is called as Tradescantia. Plasmolysis experiment was demonstrated in the plant of Tradescantia, right. Now apart from this, if you look at the process of transpiration, now when you look at the process of transpiration, you know, we talk about ascent of sap. When we talk about ascent of sap, what is ascent of sap? Upward movement of water and minerals is called, this is called upward movement of water and minerals, water and minerals. And ascent of sap was widely explained with the theory called cohesion tension theory, and this theory was being given by Dickson, and the theory was being given by Dickson. Dickson is a scientist who has given the theory called cohesion and tension theory. What it was being explained is nothing but ascent of sap. Ascent of sap is nothing but upward movement of water. That is what we studied in terms of transpiration. Now when we talk about transpiration, let us continue with that. So if you take the transpiration, so there is a loss of water. Loss of water whenever it happens so frequently from the surface of the leaf. Suppose if you take the plant from which there is a loss of water is there. So through stomata there is a loss of water. The loss of water itself we call it as what? Transpiration. When continuous loss of water is there, it causes wilting of leaves. It causes what? Wilting of leaves. Now wilting of leaves can be prevented. By applying Vaseline. Can be prevented by applying Vaseline. Simply on the surface of the leaves, if you keep Vaseline, on the surface of the leaves when you keep the Vaseline, then you can prevent the transpiration so that the leaves can come back to normal state. Then transpiration, whether it is happening or not, they have done the experiment using cobalt chloride. Using cobalt chloride. So I am writing here down cobalt chloride. Okay, so transpiration is happening or not, how do we come to know? We take the paper which is containing cobalt chloride and that turns into orange color. Whenever the cobalt chloride paper turns into orange color, that is an indication that yes, transpiration is happening. Yes, transpiration is happening. So whenever there is a loss of water near to that leaf or stomata, when you keep cobalt chloride paper, when we keep, because of the droplets falling on the paper, that turns into orange color. It is an indication that transpiration is taking place. Right. Now apart from this, now if you look at opening and closing of stomata, opening and storming of what you call closing of stomata. Now, better you write down directly glycolate. When you talk about opening and closing of stomata, opening and closing of stomata. So, when you talk about the phenomena of opening and closing of stomata, guys, do remember glycolate. There is one particular substance called a glycolate. A glycolate is a substance which induces, especially opening of stomata. Better what you can write is glycolate induces opening and closing of stomata, but most frequently with low concentration of carbon dioxide. With low concentration of carbon dioxide, it causes opening of stomata. It causes opening of stomata. So far you studied about God cells, H plus, K plus, Na plus and all we have studied, right? But here importantly, remember this we are going to study in respiration. There is one more substance called glycolate. Glycolate induces what? Opening and closing of stomata. Especially when their concentration is low carbon dioxide concentration, that causes what? Opening of stomata. Right. Now, apart from this, when you see the opening and closing of stomata, especially the opening and closing of stomata was being explained by the scientist Levitt. What you call K plus pump theory, H plus active transport theory, yes or no? So, studied and explained by what? Levitt. H plus or K plus pump theory explained by the scientist Levitt. Remember, he has done the experiment in case of mesophytes. That is why the type of stomata what we call photoactive stomata. That is why it is called as what? Photoactive stomata. He discussed about, studied about, observed and done experiment on photoactive stomata, but not the scotoactive stomata. Now, further if you look at what we have to remember about opening and closing. Now, in the process of opening and closing of stomata, as just now we studied, who is the scientist? Levitt. What do you call? K plus theory, or we call H plus active transport theory, right. So, when you take here, I am talking about opening and closing of stomata. Now, in this, along with that potassium, even sodium also involved in transportation across the membrane. Guys, you remember sodium potassium transport is active transport. In cytology, in first year syllabus, in the chapter of cytology, in one of the paragraph we study active transport. Example is nothing but K plus and Na plus transport. So, that is what is called Na and K plus pump. Why you call this pump? Because that represents what? Active transport. And this was the question. So, I told you to remember, it was the statement is from cytology first year textbook, and this is very important concept appeared in both NEET as well as in case of AIIMS. So, that is first year cytology concept. Let us relate it to opening and closing of stomata. This has been related to what? Opening and closing of stomata. Now, what you have to study in this? Now, this is a membrane. Let us take this is a membrane, transmembrane, cell membrane let us take. Now, Na plus here concentration of Na plus is 14 times greater. 14 times greater. So, let us continue in extracellular fluid. Extracellular fluid means where it is present? Extracellular, outside the cell. So, this we find in case of outside the cell. So, sodium concentration is 14 times greater in extracellular fluid. Let us take this is inside the cell, this is nothing but outside the cell. Okay. Then what about the K plus here? K plus concentration is 28 to 30 times. 28 to 30 times greater in exoplasm. Exoplasm means we talk about inside. Look at the cell. This is nothing but membrane or the cell membrane, or we take the cell. Let us take this is nothing but the cell. This is totally nothing but the cell. Now what we are discussing? Inside the cell potassium ion concentration, outside the cell sodium ion concentration. So if you look at in case of sodium potassium pump, of course there are so many theories to study opening and closing of stomata. One of the theory is sodium potassium pump theory or transport theory. In that it is explained that sodium concentration is 14 times greater in extracellular fluid, means 14 times it is greater in concentration. Similarly, potassium is 28 times greater. So, when the process is happening here, whenever you get the question that, I am writing the question here. Now, the ratio between sodium and potassium, then take that the answer is 3 potassium ions with 3 sodium ions with 2 potassium ion will be the ratio with which it crosses the cell membrane. For every transportation of 3 sodium inside, there is an out transport of 2 potassium ions. The ratio between sodium and potassium across the cell membrane in this process is nothing but 3 sodium always with 2 potassium ions. So, this is very important. That is an important concept what people have explained in sodium potassium pump, right. Now, so here what makes the transportation of ions and minerals? We studied about xylem, especially xylem is involved. Xylem has vessels and tracheids, parenchyma and fiber, right. So, if you look at the xylem part here, and we can see that in case of xylem, now anatomy compulsory xylem and phloem, we have previous NEET questions. In case of xylems, if you look at xylem vessels transport more amount of water than tracheids. What is the important thing here? More amount of water is being conducted by whom? Vessels than tracheids. That is why we say vessels are more water conducting channels when compared with the tracheids. Very important. When you get the comparison between vessels and tracheids, vessels are the main water conducting channels. That is the reason why we compare between angiosperms and gymnosperms in case of anatomy, right. So, these are the few important things which we have to remember. So, apart from this, even you can write choline are also the hormones which are found in case of a specific region of the plant. Okay. So, for example, like we have no auxins, we have gibberellins, right, like that even we find a specific hormones like choline. Choline are nothing but type of hormones. They are not found in meristematic, not found anywhere, but they found only in the specific region of the plant. As per the need of the metabolism, plant will synthesize whenever they need in a specific region. They are not found in meristematic zone and not found in axillary bud, not found in lateral bud, right. Now, apart from this, this is one thing which we have to remember. Now, root elongation zone is the zone which is involved in absorption of salts, So, when you take the root zonation here, you know this, if you take, this is nothing but a meristematic zone, you know that, and this is nothing but what? Elongation zone. So whenever the salts are getting accumulated, means the salts always get accumulated with the help of a root elongation zone. They get accumulated with the help of root elongation zone. Right. So apart from this, you know that meristematic zone contains what is called as root cap. We studied this in the first year morphology, right? Now generally root cap is absent in case of hydrophytes as well as epiphytes, which you know very well. What are hydrophytes? Whenever the plant is growing in the water, they are called to be said to be hydrophytes, and epiphytes means they grow on the branches of huge plants and they grow on the surface of other plants, they are called as epiphytes. So here root cap generally it is absent in hydrophytes and epiphytes. So these are the few things which we have to remember in case of transport in plants, right. Right. Guys, let us move ahead with the next chapter. A new chapter. Let us go with the chapter photosynthesis. Okay. Let us study about the process what we study mostly in plants. Mostly in plants, preparation of food is a very important metabolic pathway. How plants prepares food means everybody can tell me that photosynthesis. Now let us study in detail about the major process what happens in the plant. It is called photosynthesis. Now when we take the photosynthesis here, now we say photosynthesis is called as a physico-chemical process. It is called a physico-chemical process. Why do we call it as physico-chemical process here? Physical means it requires physical components, physical environment. What are the physical environment components? Water, atmospheric carbon dioxide it needs. And chemical process, why do we call chemical process? In this a light energy, it is being converted into chemical energy. A light energy, it is getting converted into what? Chemical energy by using physical components like carbon dioxide and water. Now in the plant most essentially we require what is called chlorophyll, and sunlight is essential, and that lead to formation of carbohydrates with the release of water, along with that with the release of oxygen. As oxygen is being released from the plant, it is called as a life essential process. Itself is also called as a life essential process. Right. Now, so when you look at today, we know very confidently photosynthesis process is in need of carbon dioxide, water, chlorophyll, sunlight. Right. But earlier, so what are the requirements? These are the requirements. Earlier people have not discovered at a time these are the requirements. So starting from 15th or 16th century, slowly there is an application of many people, many scientists. They tried and they have done so many experiments. In each experiment they could come to know that okay, carbon dioxide is essential. They have come to know in one of the experiment. Then after one decade or few days or few centuries or few months, few years, in another part of the world, some other scientists they discovered okay, chlorophyll is essential. Chlorophyll is essential can be discovered with the study of some experiment. Later they have done another experiment by some other scientist in some other corner of the world. They discovered in some experiment sunlight is essential. So it is not at one step of discovery, it includes multiple experiments and effort of many scientists. Now let us study about introduction in terms of experiments and discoveries. Right. So what are the experiments have been organized? in the process of revealing photosynthesis, and finally what they have discovered. So, what are the requirements? What are the requirements for photosynthesis? Carbon dioxide, water and chlorophyll, including what is called sunlight. And so, in different experiments people have discovered that yes, these components are essential. Let us start with few experiments. The first one, let us talk about a variegated leaf experiment. Variegated leaf experiment. So, what is this variegated leaf experiment here? Simply they have taken the potted plant. So, they have taken a potted plant. So, let us take the drawing of bigger leaf. So, they have taken a bigger leaf. Okay, so let us take like this. Imagine younger leaves on the tip and bigger leaves at the base. Now, the half of the leaf was being covered with black paper. So, this is being covered with black paper. Half of leaf covered with black paper. So, half of the leaf here, what we find here, it is being covered with what? A black paper, that means completely closed, and half of the leaf is exposed to sunlight. So, here is nothing but sunlight. Then after 48 hours, after 72 hours, then they took the leaf and they observed under, like with the help of iodine. You know that iodine is most commonly used. So, when they have tested with the iodine solution, they dropped the iodine droplets on the leaf which is in light and on the leaf which is in dark. So, when we put the iodine droplets, iodine solution on the half of the leaf, then it become blue colored. Blue color indicates the formation, that indicates the starch formation, that means the photosynthesis has taken place. That indicates that photosynthesis has taken place. So, whenever iodine solution is dropped here, now iodine solution that was dropped in the half of the leaf that is being exposed to light, that turned into blue color. How it could turn into blue color? Because of iodine solution. Iodine solution reacts with starch and lead to the formation of blue color. That means there is a formation of starch, that indicate that photosynthesis has taken place. And the leaf which is being covered with the black, when they tested with the iodine, here no blue color, there is no blue color, means no starch formation, meaning no photosynthesis. Why? Here because sunlight is not available. As sunlight is not available, why? Because it is being covered with black paper. As it is covered with black paper, sunlight is not available. So, in dark, when iodine droplets are kept, no blue color, that means no photosynthesis. So, this variegated leaf experiment said that yes, for the process of photosynthesis, the sunlight is essential. What is essential? The sunlight is essential. Sunlight is essential for the process of photosynthesis. In dark, it was found that no photosynthesis. So, what is that experiment? Variegated leaf experiment. Let us go with the next one, a mole's half leaf experiment. The other one is mole's half leaf experiment. Okay, so this is L, moles. Now, what is this mole's half leaf experiment here? Now, again they took what is called a potted plant. They took the potted plant, and here half of the leaf kept in the a test tube, and that contains the cotton. Cotton is placed, that is being soaked in KOH solution. So they kept the arrangement of cotton, and the cotton is soaked in solution having KOH. So I will write here KOH solution. Right. So simply a pot is being taken, a pot having the leaves is taken in the experiment. In that, half of the leaf carefully with the help of test tube placed in the half of the leaf is placed inside the test tube. Half of the leaf is placed inside the test tube. Now in that, what is there? Cotton is there. Cotton is soaked in what is called KOH solution. What the KOH solution it will do here? Now KOH solution absorbs carbon dioxide. It absorbs carbon dioxide. KOH solution will take the carbon dioxide. So when carbon dioxide is completely absorbed by the KOH solution here, there is no carbon dioxide inside the test tube. Carbon dioxide is not available. Carbon dioxide is not available for half of the leaf. It is not available. Half of the leaf where inside the jar some air is there, but from the air carbon dioxide is completely sucked and absorbed by KOH. So for half of the leaf which we kept in the jar or test tube, for that no carbon dioxide. And the leaf which is outside, so there is no carbon dioxide here, and the leaf which is present outside the test tube, for that carbon dioxide is available. Now after 48 hours it is being kept in the sunlight. So sunlight is a common thing which we can find in all experiments. So after 48 hours and 72 hours of keeping that without carbon dioxide and with the carbon dioxide, then again they tested with the iodine solution. When iodine droplets are added on both outer side of the leaf and they remove the test tube and added here also iodine. Now here same, here blue color formation indicates there is a formation of starch and there is an occurrence of photosynthesis. So the half of the leaf which is being exposing to the sunlight, for the half of the leaf carbon dioxide is available, and for that blue color formation is there because of iodine solution, and that indicates there is a formation of the starch and there is occurrence of what is called photosynthesis. Whereas in the rest part of it here no blue color, even though iodine solution is kept, but no photosynthesis. No photosynthesis. So this experiment has proved, the scientist Mole is the scientist who has done the experiment. It is called Mole's half leaf experiment. The experiment conclusion part is carbon dioxide is essential for photosynthesis. Carbon dioxide is essential for photosynthesis. So we have done two. What are they? Sunlight is essential and carbon dioxide is essential. Now it is been continued by the working of so many other scientists. The other scientist who has studied further with many experiments comes to Joseph Priestley. So let us study about the next scientist, Joseph Priestley.
ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా, నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.